Very much the TB Alliance. I feel very much a little minnow compared with everyone else who's been presenting today, and particularly the previous two presenters. Quantum DX itself is only a small little company. We're 40-odd people. We're based up in the north of England, but we do have facilities out in South Africa, in Singapore, and also a business development function out in the States as well. As I say, we are minnows. We've been focusing on developing handheld or portable molecular technologies in order to develop assays for infectious disease testing and drug resistance susceptibility, tumour profiling and companion diagnostics using our handheld device. We don't have anything on the market at the moment, but we are quite close to market, as you'll see very shortly. We were launched about 5 or 6 years ago, and we've managed to gain quite significant funding, particularly from the UK government, the EU, and also South Africa as well. We also have a little ISO-accredited laboratory, a commercial laboratory. So our whole vision is the holy grail for molecular laboratories, taking a central molecular laboratory, taking all the processes akin to that central molecular laboratory, and putting them into a handheld device where we can actually take a sample, be it from blood, be it from sputum, urine, faeces, fresh tissue, formalin-fixed tissue, and extract the DNA from it, PCR it up, amplify it up, and then detect these various SNPs on a little nanowire biosensor. And we've licensed exclusively the use of nanowires for sequencing and also nucleic acid detection and proteins from Harvard. So basically, people have said we're the Cepheid, or the handheld Cepheid instrument. We are a little bit different from them, but it's a nice thing to be told that we may be along those lines. Our assay features, we've been very much guided by FIND in this respect, is that we're trying to put it into handheld so it can be put into a pocket, used by technicians, other health professionals, as well as clinicians and nurses as well. Very, very simple to use. You literally take a disposable cartridge that's disease specific, pop the sample in, pop the disposable cartridge into the device, and press go, and out comes the results. So very easy indeed. Highly robust. We spent a lot of time and effort on making sure that it's like smartphone technology. It can potentially be dropped. It can have external noises and such like, and it won't be interfered with. Very affordable. Because we're actually not VC backed, we've been going down more the humanitarian route on looking at various health challenges. And so we're aiming for the $10 to $20 disposable cartridge cost. The actual handheld device itself is probably going to be around smartphone cost, around $500 US. But importantly, it's sample to result in under 15 minutes. We're currently developing a malaria assay, which we've got down to 10 minutes. And that's speciation as well as drug resistance status. With TB, as we all know, it's a lot more difficult with the sputum disruption, but we're probably going to be able to get around the 15-minute mark with the technologies that we have in development. As I said, we can use different sample matrices. We can do sputum, but there's obviously a few markers that could be bloodborne as well, so we can use blood, urine, and also tissue as well. So it's a highly flexible platform. We have developed it, so it has a battery life of around 8 hours, and its mains as well. We haven't got to the solar power yet. We're only a small company, we can only do one thing at a time, but it does have a battery life. And the throughput on the battery will be around 30 samples on an individual instrument. We've also got a docking station attached to it as well, so you can actually put around either 4 to 6 instruments on the docking station, so you can do multiple samples at the same time. And it's highly flexible. The actual little nanowire biosensor we use can be arrayed in different ways. So we can put, at the moment we've got around 50 different markers on. We can do many more than that, around 1,000. And with a bit more design, we can probably do 10,000 different features, all concurrently. So we've also been future-proofing the device, so not only are we doing an array or SNP array, we're also adding in later on sequencing by synthesis, and then later on you'll see, I'll tell you a little bit about it, the long read length analysis as well, which we believe is very important for drug resistant status. Very like the Elaine, we've had a look at, when we've been down in South Africa, we've had a look at what's been around in terms of sputum pots, and we've actually developed, I can't really show it at the moment, it's quite commercially sensitive, but we've developed an ability to put those pots and port them onto our actual cassette, disposable cassette. We're able to harvest and concentrate very low titer mycobacterium, and there's on-cartridge reception, disruption and lysis, so there's no pre-prep stage. We have developed a very rapid DNA extraction purification stage using a proprietary filter where literally the sample goes in and DNA comes out. So turned general extraction on its head. And when you compare us to the QIAGEN gold standard, on the far right-hand side, you'll actually see our mechanical lysis, along with our DNA extraction Q filter, actually provides better or improved results to the QIAGEN gold standard in terms of DNA extraction. Likewise, we must get on board reagents. We use lyophilized reagents. We scoured the world all over the place. We've got a couple of companies that we license lyophilized reagents to, different PCRs and such like. So as a sample comes through the device, it will then mix with the lyophilized reagents, and the reactions happen very quickly. This is an old continuous flow PCR we use. We've actually got it down to about a euro-sized coin, and it's planar, it's very flat. But just to give you an idea that we have 3 heating sections, and there's the wires or the tube go round it, and it heats and cools, and we get around 30 cycles in around 4 minutes using this particular technology. And it's just data to show that we get great results. Then our Nanowire FET biosensor is literally rows and rows and rows and rows of different nanowires, and we're able to print particular target sequences onto the nanowires very easily using a type of inkjet printer. And then at the moment we use validated biomarkers, we can print those on, but in terms of flexibility in the future, we're able to take new validated biomarkers very quickly and print them on. The technology, little biosensor, is very, very cheap indeed. It costs a matter of cents, and it's very easy to print on as well. And we use electrical impedance of the nanowires to detect the DNA's innate electrical discharge. And that gets converted from binary code, so it's a 1 if there is a particular SNP present, or it's a 0 if that SNP isn't present. And then we have ASICs underneath that to filter out some of the background sound. And then we have a QNX system, which is an FDA-approved system, software system, where we're able to translate the binary code into traditional text, and out it comes actually onto the display. So what you see at the moment in our device is that as the sample washes over, this is a 64-channel nanowire biosensor, you'll actually see the reading on the handheld actually shows a reaction. A particular SNP is present, and it's quite clear. So that gets translated into text, and when you actually use the result, you reuse the handheld, you won't be seeing that, you'll actually be seeing text instead. We're very, very keen on the drug resistant status, and we believe that whilst you can have lots and lots of SNPs around, the best way to actually look at drug resistant status is to be able to sequence it. So alongside this, it's at a much earlier stage than the SNP analysis, is our sequencing technology, where literally we are taking a cell, breaking it open, and then translocating the DNA through a nanochannel, underneath which are hundreds, if not thousands, or millions of our nanowire biosensors, literally reading the strand of DNA as it goes across. So we're reading every single molecule, every single base pair, not once like nanopore technology, but many tens of thousands of times. So it's highly accurate. And it's looking very promising at the moment, picking up most molecules, and it's ideal for long terminal repeats and such like. So things like wanting to develop a TB and an HIV assay alongside, this type of technology will be ideal to be able to sequence millions of genomes in seconds, and for very, very low cost as well. So I have to say thank you to everyone, all our different partners. We have global partners all around the world, but thank you also to Find for guiding us down the route and the direction of our company.